Good morning, everyone. I am Dr. Navishya from School of Agriculture, SR University. Today, we are going to discuss about photosynthesis and strategies for improving the photosynthesis. So, these are the contents we are going to cover in this lecture. What do you mean by photosynthesis and cyclic and non-cyclic photophosphorylation? One by one, I will explain in detail. And Kelvin cycle and C4 cycle at the end. Strategies for improving photosynthetic efficiency. Let's start with the small introduction. Photosynthesis means it is the process by which green plants converts light energy into chemical energy. So usually green plant is having green color of chlorophyll content. What it will do means using the carbon dioxide and water in the presence of sunlight, this carbon dioxide fixed into a glucose, and during the process from water it releases oxygen. So this is known as conversion of light energy into chemical energy, that is known as photosynthesis. And site of photosynthesis is chloroplast. The cell is made up of lot of cell organelles is there. In chloroplast photosynthesis takes place. So photosynthesis are categorized into two categories. One is light reaction and another one is dark reaction. So even though it takes place during the daytime, we call it as a dark reaction because it is a light independent reaction. One is light dependent reaction. That one we call it as a light reaction. Another one is light independent reaction, that is called as a dark reaction. So it is also known as the Hill reaction, and another one is Blackman's reaction based on the scientist. One more thing you need to remember is where it will take place. In chloroplast, we have two categories. One is the ring structures are there in the chloroplast. We call it as the thylakoids, and these ring structures are surrounded by the some matrix material. We call it as a stroma. In the thylakoids, light reaction takes place, and in stroma matrix region, dark reaction or carbon dioxide fixation reaction will take place. So coming to non-cyclic photophosphorylation. So light reaction, we have again two categories. One is non-cyclic photophosphorylation and another one is cyclic photophosphorylation. So why we call it as a non-cyclic and cyclic means there are two photosystems are there in during the light reaction. These two photosystems are present on the thylakoid membranes. So once it will take the electrons energy from the sunlight, the electrons will excite from this photosystem to absorb 680 nanometer radiation, and photosystem 1 absorb 700 nanometer of radiation. So once it excited, these electrons will transfer through series of intermediate compound and it never come back to the photosystem 2. That's why we call it as a non-cyclic photophosphorylation. In cyclic, it is come back to the photosystem 2. So what happens once the light comes? It excites and later pheophytin, this one is plastoquinone, and later cytochrome B6F complex, at the end plastocyanin. All these are the intermediates to transfer the electrons, and later electrons transfer to the photosystem 1 by plastocyanin. During this transfer of electrons inside cytochrome B6 complex, once the electrons is circulate, so the ATP is generated in the cytochrome B6F complex. Later at the end, after exciting photosystem 1, the electrons transfer through iron sulfur clusters, later ferredoxin, and at the end it will reduce the NADP plus to NADPH. So end product of this lighter reaction means one is ATP in the cytochrome B6F complex, and at the end NADPH. These are the energy compounds which are released at the end of light reaction. And in cyclic photophosphorylation, what happens means only photosystem 1 is involved in cyclic photophosphorylation. Here, after getting energy from the sun, the electrons are excited, and after exciting, directly electrons, instead of transferring to the pyridoxine, the electrons will transfer to the plastoquinone and then cytochrome B6F complex, and then photosystem 1. So it will takes in a cycle manner. That's why we call it as a cyclic photophosphorylation. So here the difference between non-cyclic and cyclic means only ATP is produced during the cyclic photophosphorylation, but in non-cyclic at the end we have both ATP and NADPH energy molecules are released from the non-cyclic photophosphorylation. So it will release in a 1:1 ratio. So here only ATP is released in the cyclic photophosphorylation. So coming to carbon fixation reactions, after getting energy from the light reactions, the plant will fix the carbon dioxide. So we have several carbon dioxide pathways, C3 pathway, C4 pathway and CAM pathway. So C3, C4, CAM, these are not takes place in a single plant. Based on the specificity, we have names like C3, C4 and CAM. There are different plants and different pathways are there. Coming to Kelvin cycle. Kelvin cycle is nothing but it's a basic cycle, C3 cycle. So Kelvin cycle takes place in the three steps. One is carboxylation, reduction, and another one is regeneration. So carboxylation means it is the initial step. First acceptor of this carbon dioxide is one enzyme is the Rubisco. It is the most abundant enzyme. So we also call it as a RUBP carboxylase. So this enzyme what it will do, it's a 5 carbon compound. So this 5 carbon enzyme, once it accepts the carbon dioxide, it will become unstable. So it will go split because any enzymes are made up of proteins. After splitting, and it will produce a 3-phosphoglycerate. This 3-phosphoglycerate is the 3 carbon compound from 5 carbon plus 1 carbon, 6 carbon, and once it split because after carboxylation it produces 2 molecules of 3 carbon compound, that is known as 3-phosphoglycerate. So once 3-phosphoglycerate is produced, this will utilize ATP and NADPH. We already discussed this ATP and NADPH is producing from the light reaction, that to non-cyclic pathway. After that, utilizing this energy, so 3-phosphoglycerate will undergo reduction and produces glyceroldehyde 3-phosphate. It is the reduced molecule. This glyceroldehyde 3-phosphate, for example, if it is go for 6 carbon dioxide, 12 phosphoglycerate will produce. After that, among the two will go for the glucose synthesis or sucrose synthesis, and remaining will enter the regeneration pathway. In regeneration pathway also it is need energy to regenerate this enzyme RUBP compound. To regenerate this compound ribulose 1,5-bisphosphate compound, we need again extra energy. This energy is provided by cyclic photophosphorylation. So we have three cycles. So sorry, in this cycle we have three steps. One is carboxylation, and another one is reduction, and another one is regeneration. Carboxylation means accepting the carbon dioxide. Reduction means carbon dioxide after splitting 3-phosphoglycerate will reduce to glyceroldehyde 3-phosphate. After that, from this glyceroldehyde 3-phosphate, the RUBP will regenerate. So this is the basic cycle. So it is very complex pathway. Just this one is very simple. All these steps will take place in the Kelvin cycle. First, after this 3-phosphoglycerate, it will come back as again Rubisco through this intermediates. Lot of intermediates will come. So no need to muck up. Just basic step is try to understand carboxylation, reduction, and regeneration. So at the end, we will get 3 ATP and 2 NADP molecules are required to fix the 1 molecule of carbon dioxide in C3 plant. This is highly important. In most of the exams, they will directly ask that how many ATP and NADP is required to fix the 1 carbon dioxide in the plant. You need to mention as a 3 ATP is required and 2 NADP is required. You need to mention because it varies from plant to plant. So how it is requiring? First is ATP and NADP is required, and extra ATP is required for regeneration. That's why more number of ATP we require and 2 NADP is required to fix the carbon dioxide. Coming to C4 cycle, what is the difference between C3 and C4 means in the leaf anatomy only you can categorize that. Here the cell structure is different, this cell structure is different. So in the C4 leaves, we will call it as a Kranz anatomy. What do you mean by Kranz anatomy means? For example, this is the bundle sheath cells. Bundle sheath cells means xylem and phloem. Xylem and phloem surrounded by the sheet of cells that is in a circular manner, ring-like structure. So this ring-like structure surrounded by another ring formation of mesophyll cells. They are orally arranged in a ring format. That ring structure is known as Kranz anatomy. This ring structure it won't be available in the C3 plant. What is the advantage of this Kranz anatomy? I will discuss with you later. First, try to understand that there is a structural difference between the C3 and C4, and later we will discuss that why C4 plant is more efficient photosynthetically compared to C3. I will discuss with you. So one major difference is Kranz anatomy. Another major difference in C3 and C4 cycle is C3 cycle or Kelvin cycle takes place only in the mesophyll cells. It complete entire process, that is carboxylation, reduction, regeneration. All the process will complete in the C3 cycle. And in C4 cycle, what happens means two cells are present. One is mesophyll cell is present, another one is bundle sheath cell is present. Why? Because in the C3 cycle, only one enzyme is involved, that is rubisco. Rubisco is present in the mesophyll cells. Here, two enzymes are involved. Here, rubisco is present in the bundle sheath cells, not in the mesophyll cells. In mesophyll cells, we have another enzyme is there, that is phosphoenol pyruvate carboxylase. So if anyone asks about what is the difference between C3 and C4 means Kranz anatomy is present here, Kranz anatomy it won't present in the C3, and complete Kelvin cycle or carbon dioxide fixing it takes place in single cell. In C4 it takes place in two cells, and first acceptor of the carbon dioxide is rubisco, rubulose 1,5 bisphosphate. Here first acceptor of carbon dioxide is phosphoenol pyruvate. It is present in the mesophyll cell. In next slide I will explain in detail. So here it is the mesophyll cell, it is bundle sheath cells. So first once carbon dioxide enter, what plant will do? It will convert it into bicarbonate. Later it is accepted by PEP, phosphoenol pyruvate carboxylase. Once it is accepted, it produces OAA, oxaloacetic acid. Oxaloacetic acid is the 4 carbon compound. That's why we call it as a C4 cycle. So how the name they defined means in C3 cycle, first production of the compound is phosphoglycerate. This phosphoglycerate is the 3 carbon compound. That's why we call it as a C3 pathway. And first product, whatever stable product is produced in the C4 oxaloacetate, it is a 4 carbon compound. That's why we call it as a C4 cycle. So what happens once the oxaloacetic acid is produced? It will enter the chloroplast, and this oxaloacetic acid converted into a malic acid. So after production of the malic acid, this malic acid will go to the bundle sheaths. Here malic acid will split into pyruvate. While splitting into pyruvate, it will release as the carbon dioxide. This carbon dioxide accepted by the rubisco and later Kelvin cycle. We already discussed Kelvin cycle means that carboxylation, reduction, regeneration, all process will take place inside this bundle sheath cell. So what is the usage of this means? This rubisco is there. This enzyme have a characteristics that this rubisco will accept oxygen also and carbon dioxide also. So if it is accept carbon dioxide, it produce 2 phosphoglycerate. Instead of carbon dioxide, if rubisco accept the oxygen means what happens? One instead of two, it will produce only one phosphoglycerate and another one is glycolate it will produce. It is the energy reduction process means we lost some energy because efficiently it won't convert this carbon dioxide to glucose. Almost 25 to 30% of carbon dioxide fixation will reduce because of this reaction, that one we call it as a respiration. We already know respiration means we inhale the oxygen. Like that, the carbon dioxide accepted by the Rubisco, we call it as a carboxylation. Instead of carbon dioxide, it accepts the oxygen, means we call it as a photorespiration. In the presence of light, it takes oxygen instead of carbon dioxide. It is a wastage process. But that photorespiration, it is not there in C4. Why? Because here first cell near to the atmosphere is mesophyll cells. In these cells, there is no Rubisco is there. In these cells, PEP carboxylase enzyme is present. That's why this PEP carboxylase only accept the carbon dioxide. It won't accept oxygen. There is no direct contact of Rubisco with atmosphere. That's why PEP will accept the carbon dioxide, and this PEP converted to malate, and malate will go to the bundle sheath cells. Here in the bundle sheath cells, inside this leaf anatomy, in the bundle sheath cells, we have Rubisco. This Rubisco accepts the carbon dioxide, and later it will convert it into a sugars in the bundle shell. There is no contact of Rubisco directly with the atmosphere. That's why there is no photorespiration. Anyone ask you about the why C4 plant is more efficient than the C3, you can tell this one as an answer. Photorespiration is present in C3, that will reduce 30 to 40% of its efficiency. In this C4 plant, photorespiration is not there. Because of that reason, C4 plant is comparatively highly efficient than C3. So here what is this means? We already discussed in C3 plants, to fix one carbon dioxide we need 3 ATP and 2 NADPH. Then why in C4 plant, even though it is more efficient, it needs extra energy like 5 ATP and 2 NADPH. Where it will need extra means, see here in the Kelvin cycle we need, I will go back to understand you. So here in the Kelvin cycle, we need, here you see 2 ATP and 2 NADPH it is using. Extra ATP it is using to Rubisco. So overall 3 ATP and 2 NADPH need to do this Kelvin cycle. So but in C4, the regeneration of Rubisco need extra ATP. So 3 ATP and 2 NADP is required in bundle sheath cells. After this, Rubisco enzyme regenerate in bundle sheath cells, but this PEP carboxylase regenerate in mesophyll cells. For this regeneration of PEP, we again need ATP. After this pyruvic acid produced from the malic acid, it will come back to chloroplast. This pyruvic acid converted, regenerated into PEP. For this regeneration, we need extra ATP. So regeneration of PEP also need ATP, regeneration of Rubisco also need ATP. That's why the ATP consumption is more in the carbon dioxide fixation in the C4 plant. Coming to CAMP plant. So it is almost similar to C4, but the difference here is only one cell. Like C3 plant, there is only one cell is there, that is mesophyll cell. There is no craniotomy, there is no difference in the bundle sheath cells or mesophyll cells. Only one cell is there, that is mesophyll cell. Then what is the difference between C3 and CAMP plant means? In the CAMP plant, so the first acceptor is of course PEP, but here stomata opens during the night time and closes during the daytime. What it means? These CAMP plants majorly they are desert plants like cactus or some pineapple is also a CAMP plant. Agave is also a CAMP plant. So what they will do to avoid the excess transpiration losses, to avoid the excess energy losses, they will close the stomata during the daytime and open during the night time. So carbon dioxide enter during the night time. But we already discussed photosynthesis. Photosynthesis means synthesizing the glucose in the presence of light. So light is required for this carbon dioxide fixation. That's why the plant, how it is modified itself means, during night it will uptake carbon dioxide, and of course here also PEP carboxylase is the first acceptor, and it will store it as a four carbon compound, that is oxaloacetic acid. After this, oxaloacetic acid is same up to here, same like C4 malic acid. After this, malic acid store it transfer, instead of there is no two cell, this malic acid store in the vacuole during the night time when stomata open. So then in the morning time when stomata close, there is no diffusion of carbon dioxide. At that time what happens, this whatever the stored in the malic acid is there in the vacuole, it come out. So this malic acid enter the chloroplast. Malic acid, as we discussed, it is reduced to, or it split into, sorry, oxidized to pyruvic acid, and it releases the carbon dioxide. During this carbon dioxide, same Kelvin cycle happens. This pyruvic acid is there now, it will regenerate. It regenerate the PEP carboxylase. So the major difference between C3 C4 scam plant means, so C3 plant, it takes place in mesophyll cells, of course. C4 plants, we have two cells, that is mesophyll cell is there, and another one is bundle sheath cell is there, that is Kranz anatomy. In CAM plant, the difference is here the stomata opens during the night time and close during the daytime. So like as I told, all the three pathways not present in a single plant. Based on the plants, we have a separate classification is there. The cotton, rice, spinach, lettuce, whatever the mentioned over here, all these are C3 plants. Only Kelvin cycle takes place. In C4 plants, sorghum, maize, sugarcane, amaranthus, all these are compared to C3, they are photosynthetically highly efficient. And here we have Kranz anatomy. Here we have carbon dioxide fixation takes place in the two cells. These almost all the plants of the GAM plants, majorly, not like almost all, majorly these are all the desert plants. And pineapple we are regularly we will see. So in the pineapple is also a CAM plant. It opens tomato during the night time and closes during the daytime. So small difference means C3 plants are first acceptor is Rubisco, in C4 is PEP carboxylase, in CAM also PEP carboxylase. Here it takes place in one cell, here it takes place in two cell, here it takes place in same one cell. And another one is this, among the three, which is the highly photosynthetic efficient means C4, because of its efficiency of PEP carboxylase. In CAM plant, even though same mechanism is present, because of this stomatal conductance, stomatal conductance avoid the carbon dioxide uptake. That's why compared to this C4, it is quite less efficient, and C3 very low efficiency because of the presence of photorespiration. What do you mean by photorespiration? Means the Rubisco will accept the oxygen instead of carbon dioxide. You need to remember all these examples of C3, C4 and CAM plant. So once you understand the photosynthesis, what are all the things required for the photosynthesis? Those are all acts as limiting factors. So if anyone asks you about the factors affecting photosynthesis, you can tell any factors involved, means like whatever the raw materials required for the photosynthesis. All these are all the factors affecting photosynthesis, like light, carbon dioxide concentration. If it is more carbon dioxide present means more photosynthesis, and light is also highly important. Optimum light is required. High light intensity or exposing to the high light will also affect the flowering, and also plant will wilt. And temperature, optimum temperature is required for the enzyme activation, and water availability is essential for the process. And also chlorophyll content, chlorophyll content only absorbs the light. Of photosystem 2, photosystem 1, all these are all the protein pigment complexes. So in the photosystems, the chlorophyll is present. Not only chlorophyll, we have other pigments also. Carotenoid is there, xanthophyll, we have other pigments. All these pigments are acts as accessory pigments. Accessory pigments means these are all the pigments absorbs the radiation, but it transfer that energy to the chlorophyll molecules. And after once the chlorophyll molecule got energy, so it excites. So after exciting, it releases electrons. So we are already discussed, those electrons will pass through the electron transport intermediates. So why we need to enhance the photosynthesis is even though carbon dioxide is increasing because of the population, we need more source. So that's why we are finding way to improve the photosynthesis. We already lot of scientists work on C3 plants, because we already discussed, because of the presence of photorespiration, C3 plant efficiency is very less compared to C4. That's why scientists are working on how to improve the photosynthesis of the C3 plants. So carbon dioxide at the present almost it reached to 420 ppm of the photosynthesis. Okay, any one question will rise. So this increasing carbon dioxide, which crop is more efficient means, of course, increasing carbon dioxide increase the efficiency of C3, because if the carbon dioxide concentration is high outside, so it won't take uptake. Rubisco won't uptake the oxygen. It will uptake only the carbon dioxide. That's why increasing carbon dioxide concentration helps C3 plants more. No, not C4 plants, because anyway C4 plants won't uptake oxygen. It uptake only the carbon dioxide. That's why increasing carbon dioxide concentration will increase the efficiency of C3 plants, because C3 plants need lesser energy, 3 ATP and 2 NADPH to fix the carbon dioxide. If we increase the carbon dioxide concentration, C3 plants is the more beneficial. So to analyze that which crop is more beneficial, we have one structure is a free air carbon dioxide enrichment structure or phase structure. What they will do means, here you are seeing this large open ring structure in the field. So these ring structures will release the carbon dioxide. So in the nature, normal light and water requirement, they will increase the carbon dioxide surrounding these plants. So they will test the performance of the plants in the high carbon dioxide range. So by that results, they will find out the increasing in growth and yield, improve water use efficiency, efficiency of photosynthesis. All these they are find out that the increasing carbon dioxide is highly beneficial to C3, not much for C4, and we can avoid this photorespiration just by increasing this carbon dioxide concentration. One disadvantage is there of carbon dioxide concentration is increased, temperature is also increased, because carbon dioxide acts as a greenhouse gas. It will cause the global warming. So now move to the strategies for improving photosynthesis. Once you understand in detail about the photosynthesis, you can make your own strategies also. So where are the criteria we can improve? What are the criteria we can improve to get a higher yield? One is we already know, augmenting the carboxylation and reduction photorespiration. Repeatedly we are mentioning photorespiration, photorespiration. So to increase the photosynthesis, we need to avoid that photorespiration. How we can avoid? So to increase the number of concentration of Rubisco enzymes. If Rubisco enzymes are more, so even though it will go for the photorespiration, the efficiency is little bit increased because of number of enzymes are more. It will fix the more carbon dioxide. So next one is altering the Rubisco. Rubisco have specific site for the carbon dioxide. These are the enzymes are like proteins. Proteins are made up of amino acids. We have specific site for the carbon dioxide acceptance. If we change some specific amino acids in that site, it won't uptake the oxygen. It will, we can make it for highly specific for the carbon dioxide just by altering this Rubisco structure. And another one is introduction the carbon dioxide concentrating mechanism. Here this mechanism is already present in the so many enzymes. So they will, what they will do, they will alter their structure. They will prepare one structure carboxysome like chloroplast. They prepare one structure carboxysome. So what they will do means they will concentrate this carbon dioxide around this Rubisco by this CAM mechanism, somewhat related to CAM, not exactly CAM or C4 mechanism. They will use bicarbonate and release this carbon dioxide in around the Rubisco. That mechanism, that is carbon dioxide concentrating mechanism, whatever we discussed in C4, it also included in carbon dioxide concentrating mechanism. In bundle cells, only Rubisco get carbon dioxide, it won't get oxygen. Like that, in bacteria or in cyanobacteria, what they will do, this their carboxysome is there. In carboxysome only they will uptake or diffuse carbon dioxide. There is no entry of oxygen. That is known as carbon dioxide concentrating mechanism. And another one is there, so increasing mesophil conductance. So the carbon dioxide is a gaseous molecule, it diffuses in the membrane. You already know diffuses and osmosis, there is a difference. Diffusion means any material, solvent or solute or gaseous molecule, it move from higher concentration to lower concentration, we call it as a diffusion. Osmosis means only solvent molecules transfer through semi-permeable membrane from higher water potential to lower water potential. We call it as a osmosis. Here we are increasing mesophil conductance, means diffusion of carbon dioxide we can improve through increasing the aquaporins. Aquaporins will usually go for the water. It will helps in the uptake of the water or a transfer of water from one cell to another cell. This will also diffuse the carbon dioxide. So if we increase the aquaporins numbers on the cell membrane, we can improve the conductance of the carbon dioxide. And later modulation of photorespiratory pathway. Before understanding this, you need to understand in detail about the photorespiration. Okay, let me explain you a little bit. I don't know how much you get it. So here photorespiration means, we already discussed, Rubisco will uptake the oxygen instead of carbon dioxide. We call it as a photorespiration. So photorespiration will take place in three steps. One is chloroplast, peroxisome organelle, and later mitochondria. So here you are seeing Rubisco accept oxygen and produce one phosphoglyceric acid and another one is glycolate. Once glycolate is produced, it moves to peroxisome. Later, glycine, it will become glycine. Glycine is the amino acid. Then again, it become another amino acid serine. After that, it will reduce to hydroxypyruvate and then glyceric acid. Glyceric acid come back and again phosphoglycerate it will produce. But here you are see one carbon dioxide and also ammonia, it released in the mitochondria. So this loss of carbon dioxide will reduce the efficiency of photosynthesis. So how to avoid this? Already here you observe chloroplast, peroxisome, mitochondria. It takes place in the three organelles. So scientists what they are doing means they are inserting this COS bypass, means the glycolate oxidase, oxalate oxidase, catalase. All these three enzymes they are inserting in the chloroplast. What these enzymes will do means the suddenly they are phosphoglycolate. Once glycolate is produced, this glycolate oxidase convert this glycolate to glycolic acid. During this, it releases H2O to here in the chloroplast. H2O2 acts as a reactive oxygen species. Reactive oxygen means it is having lack of one electron. These are highly reactive. It need electrons to neutralize. So this reactive oxygen species neutralized by the antioxidants. Catalase acts as an antioxidant. That's why this one neutralized by the catalase. After that, oxalate oxidase is there. It will convert this glycolate to glycerate. So all these three enzymes, what they will do means instead of going around the three organelle, it will convert this glycolate into glycerate immediately in the chloroplast. This GOC bypass pathway helps in reducing photorespiration and also improve the photosynthesis by 12 to 30%. And later improving Rubisco regeneration. We already you already saw that lot of enzymes are there in Rubisco regeneration. So in mid kinase is there, RUBP phosphorylase is there. So all these enzymes are, for example, xylulose, eryptose, pseudoheptulose. So there are so many carbohydrates are produced during this regeneration. So all these carbohydrates conversion, there is some enzymes are there, kinase and phosphatase. So these enzymes we can improve, concentration of these enzymes we can improve to increase the RUBP regeneration. So here I mentioned some pseudoheptulase 1,7, fructose 1,6, bisphosphate, transketolase, aldolase, phosphorubilum. All these enzymes are involved in regeneration. So overexpressing this, we can improve the Rubisco content and enhance electrophotosystem 1 and photosystem 2 is there in the thylakoid membrane. These protein, pigment protein complexes content we can improve, so to improve the photosynthesis. So I think this one ends. We have some strategies to improve the photosynthesis. If you know about this photosynthesis, one is photorespiration and another one is improving photosystem 1 and 2 and another one is improving Rubisco regeneration and avoid the photorespiration by GOC bypass pathway. So we have these strategies to improve the photosynthesis, and also nowadays scientists they are doing engineering of C3 plants, converting C3 plants to C4 by inserting craniotomy or this GOC pathway or carbon concentrating mechanism in the C3 plants. So by that they are trying to improve the photosynthetic efficiency of all these plants. So as summary, so just you want to remember, I want to like to just highlight the some things. So what it means the photosynthesis is just as summary. It is a light reaction is there, dark reaction is there. In the light reaction, we will get the ATP and NADPH. In the dark reaction, we have three categories: C3, C4, and CAM pathway. So based on the plant, the carbon dioxide fixation cycle is varies, and you need to remember the enzymes involved. In C3, Rubisco enzyme is involved. In C4, we have PEP carboxylase. So photosynthesis, of course, whatever the yield is producing is because of the photosynthesis. So if you improve the photosynthesis, we can directly increase the yield content. I hope you get some knowledge regarding photosynthesis, and thank you very much.